You can see the complete
Everything is okay, yes.
Okay.
Complete full screen.
Okay, hello everybody. My name is Lago, and I'm going to tell you about part of my master thesis entitled Evaluation of Physico-Chemical Factors, Interaction in Cryopreservation of Paracentrotus lividus pluteus larvae. But first, we're going to tell you about a little bit of the cryopreservation of marine species. The cryopreservation of marine species began in 1971 with the LANAS study on onshore sperm, and 6 years later, Hatsaina and Takahashi developed the first successful cryopreservation protocol on sea urchin sperm and embryos. Since in 1990s, multitudes of cryopreservation protocols has been developed for many species and cell types, and currently there are successful protocols from embryos and larvae from the different species of echinoderms, and there are successfully protocol for embryos in Paracentrotus lividus developed by the Dr. Stefania Paredes in 2009. Paracentrotus lividus is a species with a high value, economic, ecologic, and scientific, and he has a successful protocol to guarantee improvements in the culture and facility application and a biocide and allow repopulation of overexploited stocks. In this work, we asked ourselves, is possible to modulate a stress factor such as salinity and temperature to successfully cryopreserve pluteus larvae of Paracentrotus lividus? To answer this question, we performed different salinity and temperature biocide to determine the stress tolerance rate of our larvae. And next, we performed different toxicity tests with different cryoprotectants, and the cryoprotectants that showed the best results, in a range of 0.5 molar and 3 molar, and the cryoprotectants with the best results were DMCO or dimethyl suboxide and methanol, and their LOEC was the concentration used to cryopreservation biocide, and the log is a lowest observed effect concentration. And then based on the data, the tolerance rate of our larvae, we created 4 different prior treatments, pretreatments prior to freezing. And these pretreatments, the larvae were developed during the first 48 hours of life. After these 48 hours, the larvae were cryopreserved using 2 different cryoprotectants, one with DMCO and other is methanol. In both cases, Tralose was added because the Tralose reduces the toxicity, which is very high in the case of the DMCO. The cryoprotectants were added of the larvae in a total of 15 steps and increasing the concentration of 0.1 molar per minute. Once the cryoprotectants were added, the larvae were cryopreserved in a straw following this protocol. And as a result of this work, we have a successfully cryopreserved Paracentrotus lividus larvae for the first time. And you can see the larvae in these videos, how the larvae swim inside the water. And the DMCO has been a cryoprotectant with the best result similar to the data of prior literature. Contrary to we expected, low salinity and low temperature don't show the better results and show delays on the development if you compare it with the control. And this is the treatment with showed the best results. However, although we have a successful cryopreserved or larvae, many of them show damage. And to analyze this damage, we perform a propidionioidid and Hochet fluorescence dye. And in this following picture, you can see the larvae with red dots and blue dots. The red dots are dead cells, and the blue dots are DNA inside the alive cells. And you can see the red color is more concentrated in the area of the arms, and you can see this area have more retraction of the tissue and reveal the skeleton. We don't know this damage is lethal or sublethal, and how it will affect their development. But one thing is sure, this is the first Paracentrotus lividus larvae to be successfully cryopreserved. And that's all. Thank you for your attention.
Thank you so much for such wonderful photos, and really enjoyed your talk. Congratulations. And now we have time for a few questions from anyone. So if you want to ask something, or you want to just share your opinion or your, I don't know, if you want to say a few good words to our presenter, it will be very nice. And also I want to ask all participants to look into the chat section, because here you can find absolutely wonderful comments for your talk, so just enjoy. And for now, if maybe I will just ask the first question. So you chose different pretreatment temperature to cryopreserve your larvae, and I just wonder what was driven by, what was the reasons to choose 18 and 20 degrees for specific pretreatment?
This is temperature has been determined with as previously biome site with a temperature. We evaluate all this temperature and determine this is the best temperature to the development of the larvae.
Okay, thank you so much. And also we have questions from Brian Grau. Do you have any survival data for 24 hours and for 48 hours, so for more prolonged periods?
No, evaluate the percentage of normally and the number of the larvae we survive for the moment. I need to repeat this experiment and evaluate this date.
Okay, thank you so much. And we have another question from Natalia.
Yes. Under what condition does a larva live usually? Temperature, salinity, as a normal? I repeat, under what conditions does a larva live usually in nature?
Sorry, Natalia, I don't understand.
What is the normal temperature? What is like normal natural temperature for larvae existence just in nature, and sees what is like the standard optimal temperatures for their living?
Okay, in the nature, the temperature more or less in Galicia is 15 degrees, more or less. Okay, and in our culture, we use 18 degrees.
And I have a second question. You expected that pretreatment conditions or low salinity or temperature before freezing would lead to improved cryopreservation of larvae. Why? What were your assumptions based on?
Sorry, I don't understand, Natalia.
Why do you think that pretreatment procedures will improve the cryopreservation outcome? What were your initial assumptions, and why do you actually thought that pretreatment should work?
I don't understand your question, sorry.
We are just asking what was the reason to perform this experiment to make pretreatment? Why do you think that pretreatment should improve the results of cryopreservation? What was the logic behind it?
I don't understand.
Okay, so it's absolutely
Sorry.
okay. So we understand that you wanted to improve the results. And
sorry, I will respond your question. Sorry. I think it's possible to decrease the salinity and temperature because if the salinity decreasing, you have more water inside your cell, and is most easy to dehydrate the cell. And with the temperature, if I think it's possible to use a low temperature to decreasing most better my temperature for cryopreservation the larvae.
Thank you so much.
Okay.
And the next question is from Barry Fuller.
Congratulations, Alba, on your beautiful presentation. My question is that you reported that the arms of the larvae were the areas where there was most cell death. Do you think that this is a physical effect of ice on the smaller parts of the embryo compared to the main body? Is it a physical effect of ice, do you think?
We do know in the proximal in the most experiments we determine this is a physical effect or is effect for the DMNCO, but we don't know.
Okay, thank you.
Okay.
Thank you so much for such nice presentation again, and we are running out of time, so that we need to move to another speaker. And among marine animals, not only sea urchins, they deserve a special attention, and from economic point of view, mussels are of great interest. And I would like to welcome Sarah Campos and her talk about key challenges of mussels biobanking. So Sarah, could you please share your screen and start the presentation?
Good morning, everyone. Can you hear me and see me?